All right, so chapter 9, patterns of inheritance. Do make sure that you've seen this before you start the lab because you'll kind of be lost once we get to the crosses. You'll be okay for the coin flip. So if you did want to start with that, in probability, that would be okay. But once you get to the Punnett squares, you would be a little lost without this presentation. So all right, so the big ideas are Mendel's laws. We're going to talk about variations on Mendel's laws, and we're also going to talk about the chromosomal basis of inheritance. We're going to get more into the sex chromosomes and sex-linked genes later. Right now, I want to focus on the Mendelian part of this genetics. So a little history here. Pangenesis was proposed around 400 BCE, before Christ event, or before Common Era. So depending on which nomenclature you want to go by. Hippocrates was an early explanation for inheritance, and he suggested that particles called pangenes came from all parts of the organism to be incorporated into the eggs or sperm, and that characteristics acquired during a parent's lifetime could be transferred to the offspring. So when we talk about acquired characteristics, we're talking about things like the ability to play a musical instrument, the ability to play a sport, skills that you have picked up with practice over time. So they hypothesized here, or said that, hey, if you were a great athlete, your offspring would also have that ability to be a great athlete, which is true to some extent, but they still need to practice and get good at that particular talent. So we know that's not quite true. Aristotle rejected pangenesis, arguing instead there were these particles that have the potential to produce the traits that was inherited, which is a little closer in saying that, yeah, athletes have a certain set of genetics or traits that allow them to be good athletes, whether they're a sprinter or endurance athlete. And those particular traits have the possibility of being passed on to their children as well. So they have a possibility of also having those particular traits. So the idea of heredity materials mixed in the offspring, and we have this thing called the blending hypothesis, and this kind of makes a little bit more sense in that if you look at children, they're often a blend of the parents, and so we got this idea that we had something from each parent that came together to give a child, and they were a combination of each parent. It was later rejected because it didn't explain how traits can appear in one generation, then skip a generation, and then pop up in the next generation. And Mendel was actually able to show how and why this all happens. So heredity is the transmission of traits from one generation to the next. Genetics is the scientific study of heredity. We have Gregor Mendel, who is the father of genetics. He began his research in the 1860s. He actually worked with a lot of different specimens, not just peas, but his peas are what he is most famous for. He actually relied on his background of math, physics, and chemistry to help explain this whole idea of genetics, which it's kind of interesting that a lot of people, when they read Gregor Mendel's work, did not understand it at the time. He was that far ahead of everybody else's reasoning or ability to understand that concept, that it wasn't until later that a famous mathematician actually fell upon his work, and he was able to understand it and put it in terms that we actually can understand. And I'll talk about Reginald Punnett a little later. So Mendel, he argued that parents pass on their offspring in discrete heritable factors. He stressed that these heritable factors retain their individuality generation after generation. So what he termed as factors today we call genes. A heritable feature that varies among individuals, so we talk about flower color, is called a character, and each variant of that character, such as purple or white, is a trait. That term trait can also be interchangeable. We'll use the word allele to explain what that is. So an allele for a purple flower, or you have an allele for a white flower. So when we talk about true breeding, we're talking about varieties that have been self-fertilized. Now with plants, that's easy to do because we can self-pollinate plants. So this is another reason why his research was done mostly on peas. But he would self-pollinate until they were purebred. Then he could take two purebreds, and these would be two different varieties. So maybe one was a short pea plant, one was a tall pea plant. And he'd get the pure talls and the pure shorts, which means every time they bred true. A tall would always give rise to tall peas. A short would always give rise to short peas. If we mix them, a short and a tall plant, that variety would be called a hybrid, or we'll put in the term heterozygous a little bit later. A lot of vocab in this section. So a cross-fertilization of a hybridization or genetic cross. The true breeding parental plants are referred to as the P or parental generation. The hybrid are the F1 or filial generation. Filial means children or offspring. We have that F1. When we cross the F1s, we then will get to our F2s, which would be like the grandkids. So first, when we talk about a little bit of anatomy here of a flower, which is pretty easy to look at. When we look at the anatomy of a flower, what we know here is that we have the petals, we have the receptacle here, and then we have our sepals here. But this is a flower, what we would call complete flower, and it has both male and female parts. So this right here, that is the male part that has the pollen, and then right here is the female part of the flower called the carpel. The carpel is actually made up of multiple parts, the stigma, style, and then the ovary down here, which is where the eggs are. And then we have the filament and the anther. The anther is where the pollen is produced. The pollen will then land on the sticky stigma. It will grow a pollen tube down through the style and fertilize the egg. So what we could do simply when we talk about our flowers is that he can cut off the male, and I think we show this here. So it's real easy to snip or neuter, we could say, our flowers, take off the male parts, and then we can put them in a jar, and then later we can use those with a brush and brush the pollen onto the stigmas, the sticky stigmas of the female stamens, and cross-pollinate. So he could control who fertilized who in this situation. So then, of course, we would have our mature peas in our pea pods, and then you could grow those seeds, and now we would see that we have our flowering peas. Now, it's really interesting because if we throw in some terms, dominant and recessive, we know that purple is a dominant color over white. And we know this not because we are like, oh, purple is just darker, but we actually just did the cross. We crossed the pure purple, we crossed the pure white. All the offspring showed the purple. So purple is dominant over white. It covers up the white allele. So by doing these crosses, this is what he determined. These were the dominant traits of the peas, and these are the recessive traits of the peas. So we have purple over white, axial. That's where the flowers position. So are they in the middle at the end of the stems, or are they terminal at the very tip of the branches? And then we have seed color, shape, pod shape and color, and then again, are they tall or are they short? So when we do this thing, we call it a single character or a monohybrid cross. That's where we're just looking at one trait at a time, one characteristic at a time. And so he performed all of these monohybrid crosses to determine what was dominant and what was recessive. So here we took, like I already talked about, the purple versus the white. All the F1s were purple, but here, look, the white skipped a generation, and Mendel was able to explain why this happened. So our F1s are all purple. We self-pollinate the hybrids with each other, and we end up getting 3 purples and 1 white. So these are our ratios, a 3-to-1 ratio. So all purple F1 did not. So we didn't get the blending. The blending would have said, that blending theory would have mean if you would did a purple with a white, you would have gotten something in the middle, like a light purple flower right across in the middle of them. And that's not what we got. We got that true purple, and then we explained how the white reappeared in the F2 generation. So he did this with all the other characteristics and found the same thing followed. That all of those crosses ended up getting 1 to 0, 100% in the F1 of the dominant trait. The F2s all had a 3-to-1 ratio. So he developed 4 hypotheses based on these experiments he did. 1, alleles are alternative versions of genes that account for variations in inherited characteristics. So what we're basically saying is alleles are the different forms of the gene. Is it a purple form or is it the white form? Is it the smooth form? Is it the wrinkled form? Is it the tall form or the short form? And then he realized that each characteristic that an organism has 2 alleles, one from each parent. The alleles could be the same or they could be different. If they are the same, we call them homozygous. Homo means same. If they are different, they are called heterozygous. Hetero means different. If the alleles of an inherited pair differ, then the one that determines the organism's appearance is called the dominant allele. The one that has no noticeable effect on an organism's appearance is called the recessive allele. The phenotype, so here's some words I kind of introduced a little earlier, but now I'm going to define them. The phenotype is what that organism looks like. Does that organism have a purple flower or does the organism have a white flower? The genotypes are the actual genes that flower has that gives it that phenotype. So does the purple flower have 2 dominant alleles? Is it homozygous? Or does it have a dominant and a recessive that would make it heterozygous? So that's actually what letters does that individual, or what gene does that individual have? So what we do know here is that a sperm or an egg carries only 1 allele. And we talked about this during meiosis, right? We have our divorce. We split up our 2 copies. We go from diploid to haploid. So the egg has 1 copy, the sperm has another copy. All right, and so this is the law of segregation. So before we even understood the steps of meiosis, Mendel was already saying we have the divorce. We have that separation going on. He did not know about PMAT. He didn't know about the cell cycle. He didn't know any of that stuff. He already inferred with his research that we start off with 2 copies. Our eggs and sperm go down to 1. And then when we fertilize, we go back to 2. And so Mendel's hypothesis explains that 3-to-1 ratio we saw in that F2 generation. Now, in order to understand this a little bit more clearly, Reginald Punnett, like I said, was able to come up with this nice little Punnett square, or it's like kind of like a table, or some of my students call it tic-tac-toe, to show how this happens. So here we go with our purples. Now notice the purple flower and the white flower, that's the phenotype. Now the purple flower has big P, big P. Big P, big P is the genotype. The white flower has the genotype little p, little p. Now the purple flower only has big P's to give, so all its eggs or sperm will have a big P. The white flower has only little p's to give, so all their gametes will only have a little p. When we put them together, we now have our F1 hybrid generation. And so notice it has a big P and a little p. So its genotype is big P, little p. Its phenotype is purple. It is a purple flower. But now when we go through meiosis and split up our homologous chromosomes, right? We have our divorce. Half the eggs will have a big P, the other half will have the little p. When we put them together in our little Punnett square diagram, so you can see here, we have the sperm. Half the sperm will be big P, half the sperm will be little p. We have our eggs over here. Half our eggs will be big P, half our eggs will be little p. When we put them together, so right, take that p down, this one across, we end up with our first possible. This is telling you the possible offspring. The first possible offspring I can have is a homozygous dominant big P flower. My second possible offspring is big P little p, and that would be my heterozygous. I also am going to have a heterozygous here, and then here I'm going to have my homozygous recessive. So notice that I call them my GERs, GERs. So my genotypic ratio is 1, 2, 1. 1 homozygous dominant, we always start with the homozygous dominant, 2 heterozygous, and 1 homozygous recessive. My phenotypic ratio is what I see, what is expressed. I am seeing 3 purple flowers to 1 white flower. So I have a 75% chance that my offspring will be purple. I have a 25% chance of my offspring being white. So here is where we're going to do our monohybrid crosses. What I would suggest is I would suggest watching the monohybrid video from the Amoeba Sisters. It will help because they'll move things in the boxes. It'll help you understand how to do that. And then I would go and do the monohybrid, the single cross practices. So when you go to that folder that says practice, classwork practice, go to chapter 9 and you will see I have practice for single crosses, and then I have the key, and then I have the homework, and then I have another key. So I have lots of practice. So you may not necessarily need to do the homework, but I would definitely recommend doing the practice problems and checking your work. All right, so hopefully you don't have any questions here. If you could also then go ahead and do the monohybrid cross stuff in the lab at this point in time. And then we'll pick back up with hybrid crosses.